title of this presentation is Cooperate to Compete, Identifying the Potential Role for Hippocampal Region CA2 in Episodic Memory Formation. And this title is the same as my PhD thesis. So some of you have already heard parts of the story. And it's basically the idea of what CA2 could do. And it's probably not the only function it does, and may not even turn out to be actually the function. So we haven't probably tested it yet. But the key, like let's say, just as a teaser, the idea is that CA3 and CA2 cell assemblies can somehow couple together to prioritize themselves for reactivation and therefore for memory consolidation. And a few words about myself. I'm currently a postdoc at the University in Bochum, and I also have like a mini position here in Frankfurt. And I like to start these presentations with like a personal anecdote. When I was around 10 or so, I was in an amusement park, and I sneaked into a roller coaster for which I was too small. So there was this height level, and I just passed by without everyone noticing. And then it was a very wild roller coaster and something of that kind. So it turns around, and I had the feeling that I was super loose, and I was very scared while hanging upside down in 10 meters height to fall out. And that memory really stuck. So potentially CA2 may have played a role in that. Yeah. Okay. So we approached the function of CA2 from more like, let's say, a MAR perspective. So we look at the function, at the structure of it, and there's the insights which have been published a couple of years ago. And we asked, okay, based on that kind of physiology anatomy, what could CA2 do? And just to give you a brief introduction again, and the components which are part of our ideas, and that is, so that's a classical circuit, right? Then the gyrus projects to CA3, CA3 projects to CA1. And then CA2 sits right in the middle, projecting also to CA1, but also back to CA2 to CA3. And then also like a classic study in 2014 by Kohara showed that no, actually then the gyrus also projects to CA2. And there's also strong cortical input to CA2. So like CA3 and CA2 does share their input to a large degree. However, with different strength and so on, but yeah, they get relatively similar input to some degree. And both of these regions, CA2 and CA3, they also recruit a lot of feed-forward inhibition in the region. And that is so strong, as we heard previously, that if you excite just excitatory axons onto each other's region, that the overall signal will actually quite inhibitory. So it's like a very strong feed-forward inhibition between these two regions. And then there's this lack of LTP, which we mentioned before, and it's not like CA2 is not able to express LTP. It's just that also the excitatory projections, they have specific mechanisms that prevent the excitatory plasticity. And and then there's a lot of neuromodulatory input to CA2, vasopressin, oxytocin, substance P, which can induce this plasticity. So it's not like a stiff old man who doesn't want to change anything. It's more like a diva who waits for a certain right of conditions to respond. And then there's this big inhibitory LTD, long-term depression. All these insights which Rebecca mentioned, and they are somehow not directly part of our theory. So we have to work to make that better included. Okay, this argumentation we published in a commentary in Hippocampus 2.5 years ago, and I will briefly explain that. So we have place cells in both CA3 and CA2, and as Thomas said, it's basically a cell like an animal runs on a linear track, and then certain cells are active in certain parts of the environment, and that forms kind of a sequence. And when the animal is resting, then this sequence is reactivated during so-called sharp wave ripple events, and that reactivation is crucial for memory formation. So that are basics which have been mentioned before. And there is a model for how such like encoding of a cell assembly sequence could actually happen. And that has been published by Nikolai Chenko and Henning Sprickler. in Berlin and others. And it's basically the idea that if you have pre-configured cell assemblies which are recurrently interacting, so with excitatory inhibitory interaction and recurrent interactions, that you don't need so much feed-forward potentiation to pass on the signal. So it's basically meaning that you excite the excitatory population and then it will self-amplify, and then the inhibition of each assembly will somehow dampen it again, and that signal can jump. So, and you don't need to have so much boost of a signal to make it basically reliably jump to the next assembly. And the idea could be the following, that you have an encoding phase where the animal is experiencing an environment and has basically sequentially activating of such assemblies, which we just postulate that they are organized in that way, and they're probably not discrete. We just have it discrete here for simplicity. And during consolidation, when the sequence needs to be reactivated, then by Hebbian plasticity, the connections between these assemblies have been strengthened, and that is enough to make this like sequential activation possible. So that's basically the start of our theory, and we took this idea and extended it a bit. Okay, and yeah, that's what I just said. And let me just mention that key parameters, how that work and how much feed-forward potentiation you need, is the population size and the strength of the weights and so on. And why do we think population size plays a role here? So the population size means how many cells are recruited in a given scenario. If you take a look at what number of cells are active in different scenarios, they differ a lot. It's a bit, yeah, we have never published this figure because we find it a bit hand-wavy. It's difficult to compare different studies with different recording techniques and so on. But if you, for example, focus on studies where they did the same recording in the same animals, in the same context, where animals basically run through the environment themselves, or where they observed another animal running. So for the encoding of the other animal, there are much fewer cells which are recruited, meaning that this memory trace is probably much weaker. And this means basically that reactivation of this memory trace is probably more difficult based on this model by Jedlička. Okay, and then we somehow extended it a bit to have both CA3 and CA2 here. And we took the minimal scenario we needed for our argumentation. So let's say we have two different experiences corresponding to sequences in CA3 and CA2. So we have this S0 experience with representing an experience with a lot of cell activity, for example, encoding your own movement. And then we have, other sequences S1 and S2, which represent an experience which somehow has not such strong encoding. And there we can now, by the mechanisms we just mentioned about CA2 interactions, we can change the strength of the interactions. Yeah, that's the idea. So, and now for this scenario, basically we have this two experiences now, this like Latin letter experience with strong, assemblies in CA3, and an unimportant assembly sequence in CA2, which we just don't need here because it's strong enough by itself, this S0. And we have a different experience in Greek letters, like encoded by sequences both in CA3 and CA2. And then we have, they're happening in close succession, and then sometime later, the animal needs to reactivate sequence. And basically now the question is, will it reactivate S0 or will it reactivate this S1, S2 experience? So that's the setting. And yeah, based on the Jedlička model and some not so complicated math, basically we can pose the following prediction that if S1 and S2, if they pair and if they inhibit, like if cell assemblies inhibit each other, then if there's not so much potentiation, this scenario down here, between S1 and S2, and if there's basically, as normally is prescribed, there's actually inhibition between CA2 and CA3, then the strong sequence S0, it will win because it has stronger assemblies. And with win, I mean the following scenario, we activate the first assembly in each sequence and we basically let it run through. But if there's bidirectional interaction between this S1 and S2 sequence, so bidirectional pairing of sequences in CA3 and CA2, then these sequences can join forces and overcome strong sequence. So basically this experience here in the second case can be prioritized during consolidation. So that's basically the key idea. And then we have another paper where we should publish it soon, actually, where we model that and show that actually possible using a very simple rate-based model. So we use the same idea. We extend the Chenko model by including inhibition between assemblies, and we can show that for a single assembly sequence, it just depends on the scenarios and the amount of recurrent interaction, field-court interaction, whether you get stable sequences or whether sequences just die out, or whether sequences are so overactive that they are not very distinguishable at some point anymore. So this is now one sequence, and then we extend that to two sequences. It's a very simple. It's just like some set of differential equations, quite simple, with just rate-based activations, and it's the same like population size, field-court weights, and recurrent weights are the key parameters. And with two sequences, we can actually show that we can have this kind of competition dynamics. So if we activate the first assembly in each sequence, both get active, but they will compete with each other. And if one is like a bit more powerful than the other, meaning a bit more larger, then both will suppress each other, and one will die out while the other can recover and survive. And with three sequences, we basically show that it's the same plot as before, but now with actual experiment, that if there's very little coupling between these two sequences and inhibition dominate between CA3 and CA2, that then this one sequence up here will survive, will win this strong sequence. But if they couple together, then they can actually overcome that. So it's the same argumentation. It's just showing that it actually works in a model. And one can basically tweak the model a bit and make it a bit faster so that the sequences don't slow down. That's something we should do soon. Okay, so let me just summarize that we think that the functional role of CA2 is yet to be determined, but that's some old summary, which I haven't adjusted yet for me. But basically, our argumentation is that CA2 and CA3 interactions may allow to prioritize replay of sequences, and that mechanisms may not be necessary for all kind of experiences, but especially for something which is weak by itself or needs to be prioritized in a specific way. And let me just point out that many properties of CA2, which we know by now, are not included in that. So it's just like a minimal set of what it could do. Okay, now let's come to the part where we somewhat test our own theory. And of course, we should test it. And there's a very simple prediction, which we hopefully will test at some point. It's basically, if we have two novel experiences and pair, like they're similar, let's say they're two novel linear tracks, and we pair one within release of neuromodulation, and it should work for vasopressin, oxytocin, or substance P or so, we would see that the neuromodulated experience that should have prioritized reactivation during replay. And that's an experiment to directly test or falsify our theory, which we hopefully do soon with experimental collaborators. Okay, so that has to be done, not done yet, so it's still out whether what we claim is true or not. But we have been looking at other data, and for example, Tom McHugh has actually looked in his data to at least we see some aspects of our theory already be confirmed. So part of the theory of competition is also that you suppress competing sequences. And as Tom mentioned also this morning, is that if you silence CA2 transiently by the CNO injection, that you see that the amount of co-activation of different experiences increases, meaning that there's a sharp wave ripple event and multiple experiences are activated. And now our interpretation is at least that the suppression of these events to each other doesn't work so anymore. So that means that they're co-active, and that fits very well with this idea of sequence competition. It's not it's one aspect of it, which let's say there's experimental confirmation for it. And then there's another line of evidence which at least would be coherent with our thinking, and that is if you go back to this Hilti 2014 article, which has been mentioned several times today, they had an Morris water maze task where they show basically that you don't need CA2 to learn the Morris water maze. But there were this, let's say, noteworthy deviations of early phases of learning. So let me explain the experiment here. They have 2 days where they just change the platform location, and it has a flag, so the animal just learns, okay, there is a platform, and it sees where it is, so it doesn't need to learn that much. And then you take away the flag, and you just basically hide at a fixed location for 4 trials a day for 5 days. And over time, the animal just gets better finding a platform. So path length and the latency to reach the platform goes down. And that's true both for the control animal, the YFP, and the CA2 silenced animal. And then there's a probe day, and then there is another like session where you just relocate the platform location and have again 4 trials per day for 5 days. And the animals will basically just adapt their behavior and also will be quicker and quicker to reach this platform. And then there's this this increase on the second day, which we realized, okay, that's interesting, that there seems to be an effect on learning. And they just, like Pitti and Siegelbaum, they said, okay, if you look at the whole, like doing an overall over the whole session, then it's 0.1 probability that this happens. So they noted the trend of it. And then we asked them whether we can have a closer look at this data. And we were very happy when they provided it to us. So we investigated whether we find more more evidence. And here our prediction would be the following, that especially at early learning, when you still need to, like, if you don't have so much experience, so much exposure to like an experience, that you could make use of this hippocampal, the CA2 based prioritization of memories, that the sequences you took, for example, to go to reach the platform, that they are more prominent during reactivation, and therefore learning is faster. And especially when you have when you change the platform location during reversal learning, then you have basically these old memories and the new memories. And now you somehow don't want to care about the old memories anymore, but you can't want to care about the new memories. So you should even prioritize your new experiences over thinking about the old experiences. So there also the prioritization should be even more important than during learning in the first place. Okay, with that said in mind, we looked again at their data. And they used the latency or the distance to the platform, not the latency or the path lengths, but there's many other measures. And here I just plotted one. It's the distance to the original platform. And if you take a look, of course, both groups learn. So the distance to the original platform on average decreases over time. And if you now relocate the platform, that's basically the second plot here, then at the beginning, they still swim to the old platform location, but then quickly this distance increases. And you see that consistently that CA2 silenced mice are closer to the old platform. And if you look at the occupancy plots, basically, if you look closely, you see that where it's yellow, the red dot is the new platform location, and this like dashed dotted point is the old platform location. And so CA2 silenced mice, they spend more time around the old platform location compared to the control. It's not a super strong effect. It's not like an all-in-one, but it's actually visible in the occupancy maps if you look closely. And with that measure, or for example, the fraction spend in the target quadrant, we can actually see a significant effect here. So with that in mind, we thought, okay, maybe we can actually dive even a bit more closer and look at the behavior of the animal. And if you plot the traces where the animal swims, so this is now the first 4 trials of the first day of reversal learning. And so the CA2 silenced mice, this is now like an example where you see very clearly this. Generally, I have to say it's relatively heterogeneous. It's not a clear-cut effect. So there are some CA2 silenced mice who are actually quite fast in learning and control mice or not, but on average, we see something like that. And it means basically that the CA2 silenced mice, it keeps swimming somehow to the old platform location. Even though if it has learned, like now the 3rd time it has seen, okay, there is a new platform location, it still keeps swimming to the old one. While the control mice, they're a bit faster in adapting the strategy. So they swim around and then it's not there, and then they find the new one. And for the 3rd trial here, this in this case, it directly swims to the new platform location. So we used an algorithm. You can basically feed this search traces into some specific algorithm, which has been trained on human-created classification of what this behavior is. So we can basically label this behavior. So if the animal swims only around at the edge, it's called pygmotaxis, or if it's circling, or if it takes a random path, or it's somehow systematically scanning the environment, those kind of features one can extract from it. And so there are non-spatial search strategies and there are spatial search strategies. For example, if the animal just swims directly to the target, one would call it the direct path, or if it sticks to the old platform location, it's called perseverance. So here in this plot, we plotted these different strategies for the different trials on different days. So like the upper row is the CA2 silenced mice and the lower row is the control mice. And in general, the bluish strategies are non-spatial and the greenish yellows are the spatial ones, except for this very dark one, which is the perseverance. And we didn't see any difference actually during learning, also there. But during reversal learning, if you look at the first day, you see that the amount of the perseverance is actually much more in CA2 silenced mice. And we find the not super strong there as well, but at least somewhat a significant effect. And especially when you look at the second day and the amount of non-spatial learning, that is also a lot increased in CA2 silenced mice. So it seems that even if you look closely, CA2 has potentially also a role in spatial learning. However, keep in mind that these are chronically silent mice. And as we heard before, there are chronically silenced CA2 silenced mice have these spatial seizures. So we cannot exclude by looking at this data whether maybe the seizures somehow impair learning. So it could be like an effect which has a different, trajectory in hippocampal processing. That's something with a clear limitation of reanalyzing this data, and it would have would be nice to see whether it holds also with transient silencing. Okay, the next summary is basically we don't have an experiment yet to directly falsify our claims, so I hope that we can do that at some sooner point in time. However, the suppression aspect of our theory has been confirmed, or at least there's good data speaking for it, and the strategy analysis in the Wars motor maze that fits with our ideas. However, it fits also with the idea of atomic Q and Steve Middleton, where he said that CA2 and CA3 could be like novelty encoding. Okay, and for us, the next step is actually, do we see, because this like competition idea requires that multiple sequences are activated at the same time during a sharp wave ripple. And the next step for us would be to look in existing data whether we actually find fingerprints of this competition during reactivation events. So that's something we will do. And we can do because there's a lot of data with reactivation events online. Yeah. Okay, cool. And closing this bracket with the roller coaster. So why did this memory stick more? Maybe it was a very stressful experience which led to the release of neuromodulation in CA2, which may have led to pairing of CA3, CA2 sequences and increase the replay of this event, and therefore it's such a strong memory. Maybe. Okay. With that, I want to thank you for your attention. Thanks for all the audience which stayed with us during the symposium, both here in person and virtually, and our collaborator on that, so Andrew Lair, who has been very much involved with me from the start in thinking about these ideas, and Arvind as well from KTH, and then there's Marianne Finn, Fred Hitti, Håkon Hafting, and Christian Tetzlaff, who have been also involved in different stages. Yeah, thank you very much. And now we have pizza and some drinks here, and I'm happy to answer any questions before that.